Hello everyone, this is Sharmia. In this video, we are going to see about what is meant by pathogenesis of cell injury. First of all, what is meant by cell injury? Cell injury occurs when cells are exposed to harmful stimuli such as hypoxia, ischemia, chemical agents or physical agents, infectious organism or immunological reactions. Cell injury means when our cells are exposed to harmful stimuli like hypoxia. Hypoxia means decreased oxygen supply. When our blood supply is blocked by some cholesterol or fat contents, so that the blood cannot reach the organs which it has to distribute oxygen, so that oxygen also will be reduced. Without oxygen, the cells start to die, which can lead to death of cells and injury of cells. The cell injury may be due to the both external or internal environmental causes, like infectious organism can also lead to cell injury. Immunological reactions like antibody fight against antigens, physical agents such as trauma, chemical agents like some drugs also can cause cell injury. Depending upon the severity and duration of the injury, the damage can be reversible and irreversible. Yes, depending upon the severity of the condition and duration, how many hours the stimuli was within the cell, depending upon that, the damage can be both reversible and are irreversible. Factors which are determine the cell injury means there are several kinds of factors, like first one is type, duration and severity of the injurious agents. Like the extent of the cell injury depends upon the nature, intensity and duration of the stimulus. Yes. First of all, for example, like heat means heat is a stimuli means it's the cell injury will depend upon its nature, the heat nature and intensity, whether the heat is more high or moderate or less. That is intensity and duration of the stimulus. Duration means whether 1 hour the person was exposed to heat, or 2 hours the person was exposed to heat. Based on this, the injury may reversible or irreversible. Mild injury means mostly mild injury. The heat was moderate and short duration. Within a half hour, the patient was escaped, means it is mostly reversible cell injury. But when the heat is severe, like more heat was produced or prolonged ischemia, is more prolonged heat was the patient was exposed to prolonged heat, means the cell injury will be irreversible. Like that, another one example we can say decreased blood supply. When the patient blood to coronary artery, the heart, the blood supply to heart was supplied by coronary artery. When there is a blockage in the coronary artery, the blood will not be supplied, so that oxygen also will not be supplied to heart. If the injury was mild, the block was mild and the short duration, the block was within a short duration, the block was went, and now the blood vessels was normal, means the blood supplied was normal, means the injury will be short duration, so that the heart cells will survive, reversible. But if the block was more severe and prolonged blood ischemia means blood supply was reduced for prolonged period mean it will be irreversible cell injury. This is an example. A brief interruption of the blood supply to the myocardium may cause reversible. Brief interruption means the interruption is for less period of time means it will cause a reversible injury. If the interruption was prolonged means it can lead to myocardial infarction also. Second one is type, status and adaptability of the target cell. First, the susceptibility of the cells vary according to their structure and function. Yes, based upon their different cells have different structure and different functions. Some cells have some kinds of abilities like for example skeletal muscles can tolerate hypoxia better than cardiac muscles. Hypoxia means decreased oxygen supply. When skeletal muscles has less oxygen, it can also survive for a prolonged period, but cardiac muscle cannot survive without proper oxygen for a prolonged period. Next, for example, cardiac muscles will go irreversible injury within 20 to 30 minutes of severe ischemia. Without oxygen or blood supply, the cardiac muscles start to die within 20 to 30 minutes. But skeletal muscles will not die in 20 to 30 minutes. It can survive up to 1 to 2 hours. Third one is underlying skeletal intracellular mechanism. There are three major biochemical abnormalities are involved in most forms of cell injury. In most forms of cell injury, there will be three mechanisms. First one is which is affected means mitochondria. Mitochondria is main powerhouse of the cell. Their main function is ATP production. If the mitochondria got damaged means ATP production is also reduced. There will be depletion in ATP. Next, damage to cell membrane. If there is damage to cell membranes means the permeability and transport also altered. Cell membrane main role is protection. Protecting some kinds of allowing it will allow certain compounds only from external to internal inside the cell. But in altered in cell injury condition there will be an altered cell membrane so that all external environment contents will get inside due to cell injury. Then generation of the toxic free radicals. Toxic free radicals like when the mitochondria only the ATP production will happen. By mitochondria nutrition, the electron will get from nutrition 4e minus electron. It will combine with the oxygen and the hydrogen to form H2O. It was in normal condition, but in mitochondrial damage, one electron will leak and combine with the oxygen, and it will form a superoxide radical, that is free toxic radical. Free toxic radical further damage the cell membrane and damage proteins, damage DNA, and can also damage the mitochondria. Next one is morphological consequences. Morphological consequences means due to biochemical disturbances can also produce structural changes. Structural changes like Ultrastructural changes are first of all, structural changes means what kinds of structural changes means cell swelling, plasma membrane bulbs, mitochondria also to swell, loss of microvilli. These are some kinds of structural changes which was happens in a cell injury. These changes will be visible only first in the electron microscope. Then it will be visible in the light microscope. Reversible injury changes appear. Yes, first of all, if the cell injury is reversible means reversible changes only first appear. Next only irreversible changes will appear. Next one is mechanisms of cell injury. Mechanisms of cell injury. This first one is depletion of ATP. Next one, mitochondrial damage. Third one is influx of intracellular calcium and loss of calcium homeostasis. Next one, accumulation of oxygen derived free radicals. Next one, damages in the membrane permeability. First one, depletion of ATP. ATP depletion means mostly usually due to the hypoxia, decreased oxygen supply, because oxygen is necessary for ATP synthesis. Next one is chemical injuries. Chemical injuries due to drugs and all, also ATP get depleted. Sources oxidative phosphorylation of ATP in the mitochondria and glycolytic pathway using. Yes, first of all, ATP is essential for what an ATP will be essential means membrane transport, protein synthesis, lipid synthesis, maintenance of cellular integrity. How ATP is normally produced means aerobic respiration, oxidative phosphorylation in mitochondria. Like oxygen will combine with the glucose and form 30 to 32 ATPs in a single cycle. But during decreased oxygen supply, hypoxia, anaerobic glycolysis is used for ATP production. Glucose will suppress to lactic acid. Glucose will be converted to lactic acid and it will produce 2 ATP, but it can produce only 2 ATP per glucose. Effects of ischemia and hypoxia: reduced oxygen supply, decreases aerobic respiration, ATP production falls rapidly. In ischemia, glucose supply is also reduced, but in ischemia condition, hypoxia, we can survive by using the glucose, but in ischemia, both the glucose supply also reduced and oxygen supply also get reduced, making injury more severe. Metabolic waste fibrates all accumulates in the cells. Certain cells depends injury mainly on aerobic metabolism and are highly susceptible to some cardiac muscles, neurons, renal tubular epithelius. This will mostly depend upon the aerobic metabolism, so that without oxygen they cannot metabolize the waste contents. Second one is mitochondrial damage. Mitochondria are important targets for all types of injury, including hypoxia, toxins. First one, during cell injury, first affected organ is mitochondria. The mitochondrial changes are seen in vacuoles in the mitochondria and deposits of amorphous calcium salts in mitochondrial matrix. Mitochondria can be damaged by increases of Cytosolic calcium too is due to calcium accumulation. Mitochondria get damage, oxidative stress, breakdown of phospholipids, lipid breakdown products. Supplies ATP to the cell. Damaged by calcium influx, reactive oxygen species, radiation, oxygen deprivation, toxins, and mutations in mitochondrial genes. Consequences of mitochondrial damage: formation of the mitochondrial permeability transition pore, which leads to loss of membrane potential, failure of phosphorylation, and ATP depletion, and then necrosis. Release of cytochrome C into the cytosol that activates apoptosis. Failure of oxidative phosphorylation leads to ATP depletion and formation of reactive oxygen species. When there is increased cytosolic calcium, reactive oxygen species are repeat peroxidation. In this condition, mitochondria get injured. Then there will be increased ATP production. Mitochondrial permeability transition leads to loss of membrane potential, and due to loss of membrane potential, inability to generate ATP, the cells start to die continuously. Due to cytochrome C and other pro-apoptosis proteins, apoptosis will occur. Programmed cell death. Third one is influx of intracellular calcium and loss of calcium homeostasis. First, calcium membrane damage causes increased influx of the calcium into the cell, so that the calcium will accumulate in the mitochondria, which can lead to mitochondrial swelling, formation of phospholipid-rich amorphous dendrites, and activation of the degradative enzymes. During prolonged hypoxia, a large number of the calcium will enter into the cell, and calcium will directly accumulate in the mitochondria. Mitochondria will stop the production of the ATP. Microscopic changes while seeing in the microscope, we can see the changes like mitochondria will get swell, vacuole formation, calcium deposits in the mitochondria, which can what result will occur in this means energy production will stop immediately. Yes, during membrane damage, extracellular calcium will enter into the inside, and increased cytosolic calcium, which can activate the cellular enzymes like phospholipase, protease, endonuclease, ATPase. Phospholipids decrease the phospholipids and cause membrane damage. Protease will disrupt the membrane and cytoskeletal proteins, and which can further damage the membrane. Endonuclease will further cause damage in the nucleus. Then ATPase will reduce the production of the ATP. When increased cytosolic calcium can also lead to increased mitochondrial permeability transition, which can also reduce the ATP production. Then fourth one is accumulation of oxygen derived free radicals. One of the oxygen free radical is produced normally in small amounts and removed by defense mechanisms. But once a reactive oxygen species amount increases, this will lead to the oxidative stress. Oxidative stress can further increase the cell injury, cancer, aging, and some degenerative diseases like Alzheimer. Also, reactive oxygen species are produced by leukocytes and macrophages in inflammation. Yes, reactive oxygen species mainly caused by during there will in mitochondria oxygen electrons will be produced taken from the nutrients. This electron will 4 electron must be combined with oxygen and H2 so to form the water, but there will be a due to mitochondrial damage there will be leakage of the electron, single electron from the mitochondria, and it will combine with the oxygen and can produce the oxygen free radicals which can further damage the mitochondria, cell membrane and other. Formation of the free radical. First smoking condition. In smoking condition nitric oxide will produced in UV there will be O3 plus UV. It will also produce the OH minus free radical. Inflammation also to OH minus mitochondria also produce by use this kinds of O2 free radical can further damage the DNA. How we can remove the free radical? Antioxidants, vitamin E and A, ascorbic acid, glutathione in the cytosol. Bind to protein sensors, catalyze H2O2 will convert to O2 and H2O. Superoxide dismutase glutathione level is important in cell safety. Next, last one is defects in membrane permeability. Yes, in ischemia condition, membrane damage may be the result of ATP depletion and calcium modulated activation of phospholipases. It can also be damaged directly by certain bacterial toxins, viral proteins, etc. Membrane damage, permeability damage, there will be an ultrastructural changes like loss of microvilli. Microvilli will increase the area of absorption inside the cell, but in membrane alteration there will be loss of microvilli, formation of the membrane bulbs because due to the attachment is failed during cell injury, so that for the binding the membrane bulbs will be formed in the membrane. Loss of intramembranous particles will get lost. Myelin formation. Myelins are composed of damaged membrane phospholipids arranged in concentric layers, and the membrane will get damaged. Due to membrane damage, the permeability will get collapsed, so that it will allow all the materials which has not to be enter into inside the cell. The biochemical mechanisms contribute to membrane damage are accelerated degradation of membrane phospholipid, mitochondrial dysfunction, cytoskeletal abnormalities, reactive oxygen species, lipid breakdown products. First, this reactive oxygen species will cause lipid peroxidation. Lipid peroxidation will further cause phospholipid loss and further cause membrane damage. Decreased oxygen will lead to decreased phospholipid synthesis, phospholipid loss, membrane damage. Cytosolic calcium, when ink get increased, phospholipids will get activated. Increased phospholipid degradation, lipid breakdown products will further cause membrane damage. Increased cytosolic calcium too will cause protease activation and lead to cytoskeletal damage and further cause membrane damage. Then pathogenesis of ischemia and hypoxia. Hypoxia and ischemia will cause ATP losses. Without oxygen, the ATP cannot be produced and will cause ATP loss, which can further reduce pH of the cytosol. Can also reduce the sodium pump and can also decrease protein synthesis. Decreased sodium pump will lead to structural and functional changes of the cell, which can lead to reversible cell damage. Yes, it's some kinds of reversible cell damage only. Hypoxia, normally, I was going to repeat it. During hypoxia or ischemia condition, ATP will get lost, so that damage, it will further damage the sodium pump, which can leads to structural and functional changes of the cell. This will be a reversible cell injury, which can be reversed after the stimuli is separated. Next one is physical injury. Physical injury means like when radiation or chemo drugs, trauma. In this condition, H2O, I want to combine and form OH minus free radical, which can cause in proliferating cells like epithelial cells, it will cause the DNA damage. Due to DNA damage, genetic will get damage and the mutation. DNA damage can also lead to inhibition of DNA replication or apoptosis, programmed cell death. In non-proliferating cells like neurons, lipid peroxidation will cause cell membrane damage and necrosis, continuous cell death. Is a physical injury. In physical injury, like radiation or chemo drugs, trauma. In this condition, H2O, by using H2O to free radical, oxygen free radical OH minus will be formed. In proliferating cells like epithelial cells, which will cause DNA damage. DNA damage can also cause genetic damage, mutation, which will affect cause mutation. It can also lead to inhibition of DNA replication and apoptosis, programmed cell death. Non-proliferating cells like neurons, there will be lipid peroxidation, cell membrane get damaged, and necrosis, continuous cell death. Okay, let us revise. In this video, what we go through on is first is pathogenesis of cell injury. First, cell is a functional and structural unit of all living organisms. How cell injury is caused? Due to the internal or external stimuli. Then we have seen about the pathogenesis, type, duration, type, status, the morphological changes and all. And then mechanism we have gone through: depletion of ATP, mitochondrial damage, influx of intracellular calcium and loss of calcium homeostasis, accumulation of oxygen-derived free radicals, defects in membrane permeability. And we have also seen the physical injury, pathogenesis of ischemic and hypoxic injury, and physical injury. First, Cell injury will be three types: reversible, irreversible, adaptation. Adaptation means it will the cell will get adapt structurally and functionally based on the stimuli. Atrophy, hypertrophy, hyperplasia, metaplasia comes under the adaptation. And cell injury, in cell injury there will be reversible. Reversible means ATP depletion, and all comes under the reduced oxidative phosphorylation. Unassist ATP depletion. Due to ATP depletion, water will get influx, which can cause cellular swelling, which can be reversed. In irreversible cell injury, the cell death will be caused. There are two types: apoptosis, necrosis. Apoptosis means a programmed cell death. Necrosis means continuous cell death. Okay, in this video we have seen this much. Thank you.